Good morning all. This is the fourth class in first chapter. In previous class we discussed about how the plants are producing oxygen, and also we discussed about maximum rate of photosynthesis. In which color we will see maximum rate of photosynthesis? There are 7 colors are present in the sun rays. Is called as VIBGYOR. In these 7 colors, we are observed that maximum rate of photosynthesis occurs in the red and blue colored lights. And also 0% of photosynthesis occurs in the green color light in sunlight. Maximum rate of photosynthesis occurs in the red and blue. And also we discussed about how the oxygen will produced and how can we prove oxygen is producing from plants in photosynthesis process. Oxygen is producing during photosynthesis in the presence of light. For this activity, we took hydrilla plants. Hydrilla plants are water plants. When we are using water plants, it is producing oxygen. These water plants are helpful for this activity for capturing oxygen in the test tube. And also you have to answer following questions. How can we prove that oxygen is liberating during the photosynthesis process? This is the 4-mark question. And also, how can you identify the gas evolved in hydrilla activity? So these 2 questions you have to write. And also today we discussed about there are 4 factors are important for photosynthesis: carbon dioxide, water, sunlight, and also chlorophyll. Sunlight is necessary to form starch in green plants. Sunlight is the energy resource for the photosynthesis process. Sunlight providing energy for photosynthesis. So with using of this energy only, sunlight energy only, plants prepare their own food. So whenever sunlight is absent, there is no process of photosynthesis in plants. So we have to do one activity for this. Take a potted plant with destarched leaves. For destarching, we have to keep the plant in the dark room for 1 week. After, you have to take that plant and arrange the one black paper to the leaf and tightly fix the one black paper or black chart to the some part of the leaf. And after, you have to take this entire setup to the sunlight. After 3-4 hours, you have to cut the leaf and test for the iodine starch. Iodine test for the starch. Whenever we are using iodine solution on the leaf, it is turns into the blue-black color. Only which part is we are not covered with black paper, it is turns into the blue-black color. And whenever we are covered some part of the leaf with black paper, it is does not turns into the blue-black color when we are using iodine solution, because of when we are covering some part of the leaf with black paper, it does not receiving sunlight, so it is not performing photosynthesis. There is no formation of starch. So whenever we are using iodine solution, it does not turns into the blue-black color. Next, once again I want to repeat our aim to prove sunlight is essential for photosynthesis. Operators: potted plant, clips, black paper, iodine solution. You have to take destarched plant, and with using of clips you have to arrange the black chart or black paper on the leaf, some of the part. and after arranging this you have to keep it into the sunlight. After 3-4 hours you have to deattach the plant leaf and test for the starch with using of iodine solution. Whenever we are pour iodine solution on the leaf, only which parts we are not covered with black paper, it is turns into the blue-black color. The part with covering, some parts we are covered with black paper, it is not turns into the blue-black color. So in this activity we are confirmed sunlight is essential for photosynthesis. Chlorophyll also essential for the photosynthesis process. Chlorophyll, it is present in plant leaf. Finding these points, scientists conduct the number of experiments. There are different kind of parts present in the plant. Some parts appear different color, reddish, yellowish and also green color parts. But all these color parts cannot carry the photosynthesis process. In animals also we are seeing different kind of colors are there. Especially parrots are appear green in color. But parrots does not perform the photosynthesis process. Whenever we are seeing green color things in animals and also another different kind of plants. In the plants, green color parts present in the leaves, they will carry the photosynthesis. But in the animal, green color animals, they did not carry the photosynthesis process. So after different kind of experiments, scientists find these points. And also in the 20th century, there are a number of scientists, example Ingenhouse, they will find the position of the chloroplast, and after in 1817, two scientists, Petlier and Cavenshaw, obtained the extract material from the green colored substance. They will obtain the green color substance from the plant leaf. They named as a chlorophyll. Chlorophyll means green leaf. Chlorophyll means green leaf. So Petlier, Cavenshaw extract the green colored substance from the plant leaves. It is named as chlorophyll. So whenever we are observing transverse section of leaf, there are different kind of parts are there. Transverse section of leaf. Just above, just observe this picture. In this picture we are seeing there are different kind of parts are there. Upper surface of the leaf we are seeing one transparent layer is there. It is called as cuticle. Upper surface of the leaf, transparent layer, it is called as cuticle. It is a protecting layer for the plant. Next, below the cuticle, one cell layer present is called as upper epidermis. Below the upper epidermis, we are seeing some cells are arranged in the particular way, and also inside of the cell we are seeing some dot-like structure. It is called as palisade parenchyma. Palisade parenchyma. In middle of the leaf, we are seeing some cells are present. These two cells we can call it as vascular bundles. In that, xylem and phloem tissues are present. Xylem present upside of, ante top of the vascular bundles are xylem, and below we are seeing phloem tissues are there. Xylem present on the phloem because of it is transporting material from the lower to higher parts of the plant. But phloem, it is transport the material from upper parts to lower parts. That's why phloem appear in lower side. Next, after that we are observing some cells are irregular arrangements are there. This we can call as spongy parenchyma. In the spongy parenchyma also we are seeing some large air cavities is there. Air cavities is also called as air spaces. These spaces are important for the gases exchange in the leaf. And also after that, after this spongy parenchyma, one cell layer present that we can call as lower epidermis. In this lower epidermis also In lower epidermis only we are seeing the stomata. One 4-like structure is there. It is called as stomata. Stomata is surrounded by two guard cells. Bean-shaped structure cells are present near the stomata is called as guard cell. Okay, these guard cells are useful for opening, closing of stomata. So after that we are seeing protecting layer lower side also, it is called as cuticle. These main parts are present in the leaf transfer section. First one cuticle, upper epidermis, polycid parenchyma, xylem, phloem, spongy parenchyma. Next, guard cells, lower epidermis, stomata and cuticle. And also we are seeing some differences and similarities also present between the spongy parenchyma and polycid parenchyma. In polycid parenchyma we are seeing regular arrangement of the cell. In spongy parenchyma, irregular arrangement of cells we are seeing. More chloroplasts are present in the polycid parenchyma. Less number of chloroplasts are present in the spongy parenchyma. There is no air spaces in the polycid parenchyma. Large air cavities are present in the spongy parenchyma. And also, whenever we are observing more green color appearance we are seeing on the plant upper surface because of polycid parenchyma present near the upper surface of the leaf. That's why when we are observing plant leaf, upper side it is appear more green in color. When we are seeing bottom of the leaf, it is appear some dim and light whitish color, green color we are seeing because of polycid parenchyma contains spongy parenchyma contains less number of chloroplast. This is the reason we are seeing more greenish color on the surface of the plant leaf. Chloroplast structure also important for knowing about photosynthesis process. Chloroplast and photosynthesis. Until 1954, we don't know about photosynthesis process. After that, Daniel I. Arnold was able to break up plant cell. So gently that whole chloroplast could be obtained that could carry through photosynthesis. Chloroplast. Structure of chloroplast. Chloroplast is a membrane structure. There are 3 membranes are present. The third layer forms stalk-like structures is called as grana. Third layer form into the grana. It is believed that the site for trapping of solar energy. So third layer converted into the grana. This grana only responsible for trapping of solar light, sunlight. And the intermediate fluid filled portion is called as stroma. Inside of grana and also between the granums we are seeing one fluid present. It is a colorless fluid. It is called as stroma. It is believed to be responsible for enzyme reaction leading to the synthetic of glucose, and we are also called as light reactions and dark reactions. Light reactions are occurs in the granum. Why? Because granum is responsible for trapping of sunlight, and stroma it is responsible for enzyme reactions. So it is responsible for dark reaction. There are 2 kind of reactions are present in the photosynthesis. One is light reactions, one is dark reaction. And also substance found in the chloroplast which is capture the sunlight are called as photosynthetic pigments. Some different kind of pigments are present in the chloroplast. It is important for capturing of sunlight. There are different kind of pigments are present in the chloroplast. These total pigments are important for glucose formation. Chlorophyll, in that we are also seeing there are different kind of chlorophylls are there. Commonly chlorophyll A present all kind of plant kingdom organisms. Okay. In Mm. Chlorophyll B, it is appear in yellow in green color. Chlorophyll A, it is appear blue and green color. Chlorophyll B appear it is in yellow and green color. So there are 250 to 400 pigment molecules are grouped to light harvesting complex. 250 to 400 pigment molecules are grouped as a light harvesting complex or photosynthetic unit on each thylakoid. So these are the important for trapping of sunlight. And also there are different kind of chloroplast functions are there. In that one, we want to discuss mainly three important points, important functions for chloroplast. One is converting of light energy to chemical energy. Second one, splitting of water molecules. And third one, reduction of carbon dioxide to carbohydrates. How many main important functions are there for chloroplast means? First one, converting light energy to chemical energy. Splitting of water molecules. And third one is reduction of carbon dioxide to carbohydrates. These are the main important points for chloroplast. Light is important for starting of photosynthesis process. This light is captured by the photosynthetic pigments present in the chloroplast. So this total reactions takes place in the chloroplast. So there are mainly two important reactions are present in the photosynthesis. One is light reaction, second one is dark reaction. In chloroplast structure, we are seeing mainly two parts. One is granum, one is stroma. Granum, third layer of the chloroplast form into the granum. It is responsible for the light reactions. And colorless fluid present in the chloroplast, it is called as a stroma. Stroma, it is important for the enzyme reactions or also dark reactions. So chloroplast contain number of photosynthetic pigments. It is important for capturing of sunlight. And there are three important points. There are three important functions is there for the chloroplast. One is converting light energy into the chemical energy, splitting of water molecules, and third one is reduction of carbon dioxide into carbohydrates. This is about chloroplast structure.